Hi everybody, it's your life science / biology teacher, Mr. Poser, and today we are truly wrapping up our 7th unit on heredity by continuing to talk a little bit more about Mendelian genetics, or rather the exceptions to the rule, or at least some of the exceptions to the rules of Mendelian genetics. You know, that each gamete gets 2 alleles, or each gene determines 1 trait, and that kind of stuff. A bunch of exceptions here that we're about to talk about. All right, so here's a question for you, and we're actually going to answer this not at the end of the video, but right towards the beginning here. So here's a fact. It says 1 in 12 men and 1 in 200 women are red-green colorblind. Why is it so much more common in men than in women? That's almost 10 times more common in men having this colorblind trait than it is in women. And in case you don't know what colorblindness is, this is what it might look like. So this is a colorblind test over here, and the idea is that you ought to differentiate between the colors red and green. And people with colorblindness, red-green colorblindness, cannot necessarily do that. And so why is it so much more common than it is in men than it is in women? That's because it is a sex-linked gene. All the genes that we talked about in our last video are what we call autosomal, meaning that they are located on an autosome, one of the other 22 sets of chromosomes besides the X and Y chromosomes. At the very end of that karyotype. So it's a gene located on either sex chromosome, X or Y in humans, and show a different inheritance pattern than genes on autosomes. So check it out. Red-green colorblindness is a X-linked recessive disorder, or an X-linked recessive gene. Okay, so check this out. If the mom is what we call a carrier, meaning that she has 1 copy of that recessive gene, she can pass it on, and her son might be affected by the colorblindness because the son only needs 1 copy of that gene, the Y chromosome doesn't count, and pass that on to a carrier daughter as well. So check this out. This is how sex-linked genes work with regards to Punnett squares. It's X-linked recessive, meaning males only need 1 copy of the colorblind gene to express it, since it's on the X chromosome, and that's what biological men have, is X chromosome and a Y chromosome. It's almost like the Y chromosome doesn't even count when it comes to X-linked disorders. Okay, so check this out. Here's how we would set up a Punnett square. We have a male who is X and Y without the colorblind gene, and if we have a female who is a carrier, meaning that they have the gene but they don't express that gene, that means it's recessive, right? So both of these, this man and this woman, are both normal-sighted, but they can have colorblind children. More specifically, they might have a colorblind boy. So check this out. 50-50, right? You can have the 50-50 chance of having a female or having a male offspring here. And if this colorblind gene is passed on to the female, then the daughter, is going to be a carrier. That she's going to have the gene for colorblindness, like mom, but not express it because it's not on both of her X chromosomes. It's recessive, okay? But check this out. If the male gets that colorblind gene, then he is actually going to be colorblind on account of the fact that he only needs 1 copy of this X chromosome, this X chromosome with the colorblind gene on it, in order to express it. The Y almost doesn't count, right? So check it out. According to our Punnett square here, these 2 parents have a 50-50 chance of getting a boy who's colorblind, okay, or a 50-50 chance of a girl who is going to be a carrier, right? So none of their girls are going to be colorblind, and there's a 50-50 chance of the boys being colorblind in their family, right? So that's how sex-linked genes work, and they follow a different inheritance pattern than what we saw in our last video. So this is one of the exceptions to Mendelian rules here. And there's a couple more that we're going to talk about in this video before we wrap up this unit. So as evidenced by our sex-linked genes here, Mendel's inheritance laws don't predict everything. There are exceptions to it, and here's another one. It's called incomplete dominance, when neither allele is completely dominant and hybrids have a phenotype between the parentals. What does that mean? Well, it's basically that if you're a heterozygote, that means you're going to have a phenotype that's somewhere in between, or an intermediate between your 2 parents' phenotypes, right? So if we go back to flowers here, okay, let's just say in these 4 o'clock flowers, if you're homozygous dominant for flower color, you have red petals. If you're homozygous recessive for flower color or for petal color, you have white petals. All right, check it out. The heterozygotes between these two, this is a regular old Punnett square here, except there's one catch. The heterozygotes here aren't going to be red. They're going to be pink, okay? Showing that the red phenotype has incomplete dominance, meaning that it's not quite all the way expressed. And pink is kind of like a mix between the dominant phenotype and the recessive phenotype. Okay, so there's an example of one of the examples of how Mendel wasn't always right, but that's okay. He's still the father of genetics, and his rules for helping us figure out how traits are passed down are still used today, and they're very important. Another exception to the rules are what we see in codominance. So inheritance of human blood types show codominance, or when two alleles each affect the phenotype in separate distinguishable ways. Okay, so check this out. In this example here, human blood type is determined by 3 different alleles. There's the A allele, the B allele, and the O allele. Okay, if you inherit a allele, that means you're going to be blood type A. If you don't inherit a B. If you inherit a B allele and a B allele, or a B allele and an O allele, you're going to be blood type B. Okay, if you inherit 2 O alleles, that's recessive, then you're going to be blood type O. Okay, so O is recessive, but A and B are codominant, meaning that, okay, check out these offspring here. If they have A and O, they're going to be blood type A. If they have B and O, they're going to be blood type B. If they have O and O, they're going to be blood type O. But check this out. Blood type AB means that you have an A allele and a B allele, and both are being expressed at the same time. Okay, so that means that their blood has A antigens, it's called, and B antigens at the same time. Okay, so if you know your blood type, you might be able to figure out, like, okay, what my parents' blood types could have been, or what my children's blood type might be based on, just based on some simple Punnett squares and some codominance that's being demonstrated here. Okay, so if we were to set this up in a Punnett square, we'd see something similar, right? If we cross AO and BO, these are the 4 different combinations that we might get in the children here. Another exception to the rule is when traits are polygenic, meaning that multiple genes contribute to one phenotype. Okay, and we're not going to work out any Punnett squares for that because it's nearly impossible. Think about it. The trait for height, right, for height in humans is determined by 180 different genes, meaning that we would have to have a Punnett square with 180 letters across. That wouldn't make any sense, right? That's impossible to try and do. Well, it's not impossible, but you get what I'm saying. Okay, so polygenic traits are another exception to Mendelian rules. And the last example that we're going to talk about today, there are many more exceptions to the rules here, but the last one that we're going to talk about today is how the environment also interacts with gene expression through diet, through light, through lifestyle, through pH, temperature, just the environment affecting phenotypes and the expression of genes. Okay, so 2 examples of how the environment is affecting genes here, or excuse me, is affecting phenotypes, right, is that in special types of reptiles, in turtles, and some kind of crocodiles, kinds of crocodiles, temperature of the egg incubation, right? So they got to lay their eggs, and they have to incubate them. They have to sit on them or just like keep them warm, right? But the temperature at which their eggs are incubated often determines sex. Okay, that means that 2 eggs might have the same chromosomes, the same sex chromosomes, to determine that if they're going to be biologically male or female, but the expression of those chromosomes might be different depending on the incubation of the temperature of incubation that they were at. Okay, which is pretty amazing if you ask me. You know, it's just like if you're incubated at a colder temperature, you might be biologically male. At a warmer temperature, you might be biologically female. It's actually true when it comes to reptiles. It's not for people, but for reptiles, it's actually true. And then finally, in special kinds of flowers, especially orchids, 2 orchids might have the same genes for their flower color. I know we're going back to flower color here again, but it's easy to understand, right? They might have the same genes for flower color, but if 2 plants with the same exact genotype for flower color are planted in different pH soils, then that means that their flower colors might be different, right? So this is an orchid here. Orchids have purple flower color, I want to say, in neutral pH soil, but then if the soil is a little more acidic, I think it's going to be more like a pink color. You might have to double check me on that. But once again, there's another exception to the Mendelian rules here. So yeah, it is pretty nifty here. So that'll be it for this unit. We're going to get into evolution in our next unit and tie all this genetic stuff together and figure out how it affects populations and how they change in our next unit. So let me know if you have any questions. See you next time.